questions. This is one of the things I sent out by Google Classroom. I was hoping that you would send them back so that we could give you feedback on whether or not this PDF annotation submission process is working before Friday, when you will do this again, except with an exam and not with a pre-exam. By the way, the presence of the pre-exam means that the pre-exam or the practice exam is going to be distributed to you at the end of class, also by Google Classroom. So this will, again, mimic what you're going to be expected to do on Friday for the real exam. And I don't have the key quite done yet, but I'll give you the practice exam at the end of class today digitally. Sometime before tomorrow, I'll post the key. So you can try the exam on your own. Then you can compare your answers to the key. If you don't understand something, those are really good questions to bring to class Wednesday for the review session. Okay. So I realized, let's see, is this genotype going to produce a band or multiple bands on the gel? One band or two bands? Okay, how do you know it's just one band? Right, homozygous. So this individual has the same genotype on their other chromosome, diploid individual. So how big is this piece of DNA? What's the size of this piece of DNA? Physical size, number of nucleotides. So we've got, let's see, 5 nucleotides on either side of this repeat, and then 80 nucleotides, or 80 repeats of 5. So 410. So that should be a single band on this gel just above the 400 base pair ladder band. Yes, please. This is a good time for questions.
If it's homozygous, shouldn't it be GATCG, or since it's homozygous?
Oh, sorry. Thank you very much. For goodness sakes. That would be GATCG. G80. You're right. Yep. So each chromosome, both of the chromosomes, homozygous, they're exactly the same length. 5 nucleotides on either side of a 400 base pair sequence that's just repeats of 5 nucleotides over and over and over again. All right. Other questions about this? So agarose gel electrophoresis without the PCR part. I'm just giving you, here's the piece of DNA. What does it look like when you electrophorese it on an agarose gel? This should look pretty familiar, since I just asked it again. Denature, anneal, and extend. Those are the 3 valid answers. And the reason I was asking you to type or to add a sticky note was to make sure that we could see those sorts of annotations you might make on an exam. This exam is going to have a variety of types of questions, including some short response like paragraph style. So it's going to be useful either to make sure you have clear handwriting if you're writing with your fingertip or a stylus, or to be able to use the the digital keypad on the tablet to type a response to make sure that we can read it. Really to make sure that Fatima can read it. That's why I wanted it either a note or typed text. If you handwrote it, it's fine. I just wanted to make sure we had a chance to double check. I realized, and again, if I do this during class and you notice, throw something at me, say something, raise your hand. I noticed I skipped some questions that I meant to go over last class, so now we're going back and talking about those questions. One of the questions I asked in the video that we discussed last class, and I ignored, was what's the size of this PCR product that's produced using the primers that we came up with last class? In other words, there's a primer circled in red. There's another primer So how big is this PCR amplicon that's going to be produced by PCR on that template molecule with those primers? How do you figure this out? What defines the size of a PCR product? It's where the primers are annealed. So let me change color here. So there's the 5' end of one primer. Where's the 5' end of the other primer? It's down here. So that's encircled in yellow then, that's the piece of DNA that's going to get copied. From the 5' end of the left primer to the 5' end down here of the primer that's on the right side of the screen pointing to the left, 5' to 3'. So then you just count how many nucleotides that is. Right. So the 5' ends of the two primers define the size of the PCR product that gets produced. $16? Okay. I saw one head nod, so that's enough. And the really important thing, which we have to go over, is the gel results. We have a polymorphism in this individual. Remember, we pointed out, circled in red there from last class, was the position of a SNP. C to G change in the nucleotide sequence of the paternal copy of the chromosome where there's a C and the maternal copy where there's a G.
I actually counted 17 nucleotides.
Was it 17?
That's what I counted.
Okay. Well, it's the number of nucleotides between the 5' ends of the two primers, regardless of what that number is. So we'll figure it out afterwards. But thank you for pointing that out. What's special about GGGCCC? That sequence. So there's a single nucleotide polymorphism in that sequence. So we have one chromosome, the paternal chromosome has CCCGGG. That's paternal. And the maternal version has this mutation in it that makes it CCGGGG. What's the question asking? What does the agarose gel look like? What do we have to do? That question number 4 down there in the bottom, what does it say? Draw agarose gel results with and without. What's small 1?
It's an enzyme that's going to cut at the single nucleotide polymorphism.
It's a restriction enzyme. So I introduced these in one of the videos to watch before class, not for today, but a couple of classes ago. So small 1 is a restriction enzyme. It's a protein that recognizes a specific DNA sequence. This is the sequence it recognizes, CCCGGG. And small 1 will recognize that double helix, so we can write in the opposite strand, just for completeness, of both of these molecules.
How do you know it recognizes that?
That is the best question to ask. I'm surprised it took somebody that long to ask, but thank you for doing so. Yeah?
So does it only identify with the bond between a C and a G? Kind of like a lock and key?
This protein reads the DNA sequence somehow, and we haven't gotten in class yet to how proteins recognize specific DNA sequences, but it does have to do with chemical bonding between the protein and the DNA sequence that it recognizes. So how do I know that small 1 recognizes CCCGGG? Does anybody have an idea? I Google small 1, and for me, the first hit that comes up is this website, New England Biolabs. Keep this in mind, because you'll need it for Friday probably, maybe. Google. So small 1, the NEB website. NEB, New England Biolabs, is a biotech company that sells restriction enzymes. It's where a lot of scientists purchase these enzymes from to digest DNA and PCR fragments. And there it is. It tells you what the restriction site is for small 1. So it recognizes CCC, GGG, and that black triangle right there, the pair of black triangles, tells you where it cuts the DNA. So small 1 recognizes CCC, GGG, it cuts right in the middle on both strands, between the C's and the G's. This is a great resource for figuring out what is the site, the sequence of DNA that a particular restriction enzyme will recognize, bind to, and cleave. So that's small 1. So which of these two will it cut? Either or both, paternal, maternal, or both? Wait a second. What happened when this mutation, the SNP, occurred? We have small 1's restriction site here, CCC, GGG on one strand. And remember from the video that restriction enzyme sites in most DNA sequences that proteins bind are palindromes. So we read the top strand from left to right. It reads CCC, GGG. And if we turn upside down and read the bottom strand from 5' to 3', it reads the same, CCC, GGG. So restriction enzymes and other DNA binding proteins often bind palindromic sequences. And what's true about the maternal copy of the chromosome? This mutation, the SNP, has made it become CCG, GGG, not a sequence that small 1 will bind to. It's a different DNA sequence. Only by 1 nucleotide, but that matters. The protein doesn't recognize this 6 nucleotides as its sequence. It's different.
So it would just copy the paternal?
So this enzyme will only cut right there, as we saw on the NEB website, in between the C's and the G's. It will only cut the paternal version of this chromosome. It won't cut mom's copy or the other copy of the chromosome in the cell. So how does this affect the gel? So here's an agarose gel. There's a well. So how big is mom's? After adding, so we're going to look at this plus with small 1 and minus without. So we take this whole DNA sample. Right? One individual has 2 copies of the chromosome, the paternal version and the maternal version. So we take that one person's set of chromosomes, both of them, stick them in the gel either with or without digest by small 1. So if you don't add this enzyme, what band size do we have?
If you don't add the enzyme?
Right, minus. No enzyme. The fragments are 6 nucleotides long, so we'll have one band that's 6 nucleotides. Because we haven't added the enzyme, nothing gets cut. So we have 2 molecules, one here and one here, right? The size of the DNA product doesn't differ depending on the fact that there's this mutation. Mutation hasn't changed the number of nucleotides. So it hasn't changed the size of the piece of DNA, just what it says, CCG or CCC. We can't tell the difference in DNA sequences on an agarose gel. We just look at size. So what happens when we add this enzyme?
It's going to cut the paternal strand, so it's going to go further down the gel.
So the paternal strand is going to get cut, as we looked at right here, in between. So you're going to get 2 chunks of 3 nucleotides. And that's from the paternal molecule of DNA. Okay? Please.
In that we have 2 strands of 3 nucleotides. We don't have to draw the other strand?
Which other strand? The one that
So
did?
like,
The one
I
that didn't get
expected-
cut?
Yeah, because I expected to see bands here, yeah.
You're right.
Yeah, I was gonna have something similar. Does it like clump as it goes further down the gel, or?
So we've got the maternal, the uncut version of the chromosome is still there. The mutant version, the one that had the SNP, the one that abolished, that broke the restriction site. So you add small 1, the enzyme, that chromosome doesn't get cut. So you still have one 6-nucleotide band. That's the one from mom, the maternal version. And then down here, the paternal version is the one that got cut.
So if it does get cut, you have 2 3-nucleotide strands.
Indeed.
That's only one band, so like, do they just kind of aggregate?
It's a great question. Why did I only show one band here when there are 2 pieces of DNA? They're the same size. And the only thing that the agarose del does is separate bands by size. So we have 2 bands the same size. They show up at the same location, top to bottom on the gel. What you might expect to see, and this is an excellent point, so I'm glad you made it, was that when you think about total numbers of molecules, how many molecules, if we're just looking at, we started with 2 chromosomes, let's say. There's one, there's the other. So if that is a band that represents a single chromosome, how would you tell that that was 2 pieces of DNA that are 3 nucleotides long? In an actual agarose gel, that will appear darker or brighter because there's twice as much DNA at that position. There are 2 molecules of DNA, not 1. So that's often how you might get a hint, a suspicion, that there's been a cleavage and that you have twice as many molecules of the same size. There's twice as much DNA there at that position on the gel.
We can go ahead and extract that DNA and like run further tests on it as well.
If you like. We don't need to, but you could. So let's see. Given that the darker band represents more DNA, the lighter band less DNA, what else is now wrong with this gel? How should I change it?
You do a ladder.
Another good point, separate, just talking about band intensities, how dark they are.
The one without enzyme should be darker too.
Right. This band is 2 molecules of DNA also. It's 2 chromosomes, the 2 that are shown in the upper right. So it should be twice as dark. So that's 2 molecules of 6 nucleotides. That's 2 molecules of 3 nucleotides. That's 1 molecule. And yes, absolutely, I should add a DNA ladder. That's how I would know how big these fragments are. I gave you a very ambiguous exercise to think about, at least for today. Again, having to do with this allele frequency, microsatellite size worksheet. What was wrong? Why should you be concerned about implicating that individual B has the genotype that matches the forensic sample that police are going to say, this is the guilty suspect, Your Honor? Why should we be concerned?
Chance mutation?
So, chance mutation. There's always the chance of mutations. We should always be concerned about that. That somebody looks like somebody else by chance. Wait, oh, I just gave it away. Go ahead.
No, and say, what about having one of the most common genotypes?
Right. So that is an excellent way to put a concern. How common are these genotypes and how likely it is by chance we've found a suspect that we put in a lineup, say, that by chance happens to have the same genotype as the person that left the DNA at the crime scene. So, and this is not entirely the most accurate way to do this. We'd want to look at the genotype frequencies of an entire population, like we've seen in the tables that are published that I put up. I think it was US Caucasian allele frequencies, the different microsatellites that the FBI uses to genotype us when they get our DNA. But here we have empirical data on the right there. We've got the genotypes of 8 different individuals. So how many GA3s are there? And how many GA5s?
There's 6 GA3s and 1 GA5.
So not the genotypes, but actual alleles. So everything on each, so I'd count this, for example, as that's 1 GA3, that's a second GA3, third, fourth, fifth, sixth. So there are 12 GA3s, right? We want to know how many molecules of DNA in the population have each of these allele sizes. There are 12 GA3s. How many individuals are there? 8. So there must be 16 total alleles, 2 in each of the 8 individuals. So that must mean that there are 4 GA5s. What about TC3 versus TC4? How many TC3s are there?
There's 8.
8. Must mean that there are also 8
TC4s.
TC4s. Also totals 16. And then AT2 and AT5.
10 AT2s.
And 6 AT5s?
6 AT5s.
Okay. So what's the frequency of GA3? How many GA3s, how many copies of the chromosome that just have 3 GAs, GA, 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 how many of those are there in this population that we're looking at? The 8 individuals that might be guilty. They're in the lineup. So 3/4 of the individuals we're looking at have GA3. Right, that's up here in red. That's the genotype we're looking for. We're looking for GA3, TC4s, and AT5s. So GA3s, we've got 3/4 of the people in the population have that allele. It's pretty common. Would you like to have a 75% chance that you get thrown in prison because you match somebody else's DNA? It's pretty bad odds. I wouldn't take that bet. Okay, what about the next one? Do we do any better at the TC3, TC4? So TC4 is the genotype of the sample at the crime scene. We've got a 1 and 2. 8 out of 16 total alleles are TC4. That's also pretty bad odds. What about AT5? 6 out of 16, 3/5. So what's the probability that any one person in this fictional world has the genotype that left the DNA at the crime scene? It's 3/4 times 1/2 times 3/5. Pardon?
Isn't it 3/8? Or
Yeah.
8/5?
Sorry, AT5, you're right. Not 3 out of 5. It's 6 out of 16, which is 3/8.
Oh, okay.
Thank you. So 3/4 times 1/2 times 3 out of 8. It's approximately 1 out of 7. It's approximately a 7th. Which means that 1 out of 7 individuals by chance would have that genotype. The one in the upper left, the one that was left at the crime scene. 1 out of 7 individuals in that world, at least among the 8 people that are suspects, by chance, 1 of 7. We looked at 8 suspects, so how many of them by chance should have a matching genotype? 1 point something. 1 in 7 chance of having a matching genotype. We look at 8 people. At least one of them odds are, will have that same genotype, is what this means. Even though they have a matching genotype, we can't say, Your Honor, they did it. Because that individual B, in the police lineup, just happens to have a very common genotype that happens to be the common genotype that was left at the crime scene. So you need to know something about how common and how rare alleles are before you start telling people that they're guilty, or at least alleging that people are guilty. So an important point, calculate allele frequencies, you multiply them together to estimate for the number of alleles, for the number of loci you're looking at, how likely it is that somebody matches a DNA sample. And one more point before I get to your question, but I will. What does this mean about DNA fingerprinting? What's an implication? What's the best practice for DNA fingerprinting?
You need some other kind of evidence to back it up.
Well, okay, so that's a fair point. You need some sort of other evidence beyond just DNA samples.
It's not always 100%, correct?
So DNA testing is not always 100% accurate.
You should use as big as a sample, like use as many pairs as you possibly can.
Yes. And that's why the CODIS index looks at 23 different microsatellites, not just 3. The more different spots on different chromosomes you look at, the less and less likely it is by chance that you find 2 people that match. Because you're multiplying fractions. Right? Here we multiplied 3 fractions and got a relatively large number still, 1 in 7 chance of people matching at random. You do that 23 times, if it's a half times a half times a half, making up the number half. If all alleles were at 50% frequency, every allele was just as likely to be in every other person on the planet. If you look at 23 of them, that's 1/2 to the 23rd power. It's a really tiny chance that any 2 people would share the same 23 locus genotype at the same time. And that's the bottom line. We're going to look at more loci to genotype people if you're trying to do ancestry or forensics. Sir?
You answered that question.
Ah, fantastic. So just so you understand that I'm not just picking on US Caucasians, I thought I should probably show you the allele frequency tables for a couple other populations that actually exist from this paper. I know it's pretty small. Hopefully you've got it copied on your tablet screens there. But I want to point out a couple of patterns. So look at your device, or look at the screen up here if it's not too small. We've got Caucasian, Hispanic, and African American, independent of sex. Are there any differences? Are they significant, or not significant? Are they meaningful differences in allele frequencies? So looking at the first one, CSF1P0 allele 8. Or let's look at allele 9. It's in 1.2% of US Caucasians, 2.1% of US Hispanics, and 3.7% of US African Americans. Not huge differences in frequency, like 1 in 90 people, 1 in 95 people, something like that. They're pretty rare. But there are some up here that can be quite different between US populations. And I challenge you, or that's not a challenge, I offer the opportunity to investigate which of these markers are most diagnostic of heritage. Which ones are the most different between different populations? An important take-home message, I think, though, from an anthropological point of view, is that we're all really similar. Most of these frequencies aren't that different. Hispanic, African American, Caucasian, you name it. there are very similar general patterns. There are just often just nuances in how many of different populations have different sizes of alleles. So we're all very similar genetically, subtly different. Bless you. I wonder if sneezing is heritable. Hopefully it's not infectious. All right, let's look briefly at Socrative. Ooh. Videos for today, bioinformatics. Bless you again. Let's see. Number of characters, period, greater than, less than, pound sign, or hashtag for this generation. And the @ symbol. Which character starts a FASTA formatted sequence? Nice work. That's correct. The 79% of you had it right. That symbol. So what follows that symbol in a FASTA formatted sequence? Pardon? We just need some sort of name of the sequence. So this is not DNA sequence. You start each line with the symbol, and then you give that sequence a name. My favorite sequence, or whatever. Then there's a character return, or a paragraph mark. And then on the next line, you start the actual DNA sequence that you're interested in studying. So that's a FASTA formatted sequence. And the only thing this is it's arbitrary. The character that's used, this is just a convention of geneticists. We needed something to know that we would all write DNA sequences in the same format so that when we use these web-based tools or computational sequence analysis tools, they all expect the DNA sequence to be provided to them in the same format. So it's just a format. There's nothing special about the greater than symbol. Or is that the less than symbol? I can never remember which was which.
That's great.
Okay, thanks. It's been a long time since math. Okay. So you didn't Google this because Google says, I don't know what the hell this is. Too many characters. What is this? Homo sapiens. And this gene, the DNA sequence is from the Y chromosome. Which gene? It's a gene called SRY. It's the gene that makes males males, which is why it's only found on the Y chromosome. You get a Y chromosome, you have this gene. If you have this gene, this is the gene that tells the body to develop as a male body. We'll maybe look at it later in the semester in more detail. You ladies are women because you don't have SRY. If you did, you'd be males. That's how it works. How would we make this a FASTA formatted sequence? I just gave you a DNA sequence. It wasn't formatted properly. You can copy and paste this into NCBI, which is what you should have done to figure out what gene this was. And I'll show you a couple of screenshots in case you didn't get that far. But how would we make this a FASTA sequence? Yep, so right above, the line above the sequence, we'd have that symbol, and then some sort of name. Don't know WTF this is. And if you're going to use a DNA sequence analysis tool and you stick that in, it would process that, and then at the end, when it shows you the web page with the results, it would say, DNA sequence, I don't know WTF this is, and then it would give you the rest of the results. So it's a name you give the sequence to help you remember what analysis it was you were doing with that web-based program.
So the FASTA is used to literally name the sequence that you're working with?
Exactly. That's the whole point of FASTA sequence. You just give your sequence a name. That's the only point. So when you successfully copy and paste that sequence into the BLAST website, which if you don't know what I'm talking about, watch the video. You'll get a window that looks something like that. Part of the window anyway, will say sequences producing significant alignments. And this is basically the purpose, again, just to reiterate really briefly, BLAST takes your sequence that you feed into the program and it compares it to the database of all known DNA sequences. It just looks for sequence matches. So if you type in ATGC and say, here, NCBI database, look for every spot in every organism that has an ATCG, then it will spit out a list of, this is all of the times ATCG is found on any gene and any chromosome in every organism that's in the database, which is billions and trillions. So don't use 4 nucleotide DNA sequences. But if you do something like stick in a sequence that's long, if it actually came from an organism that's DNA has been sequenced, then it will tell you, oh, this sequence that you pasted in matches Homo sapiens, Homo sapiens, Homo sapiens, but you get a little bit farther down the list, you'll start seeing this DNA sequence also matches things like gorillas and chimpanzees, but it's always the same gene. SRY, SRY, from the Y chromosome, SRY, SRY. Yeah?
I don't know if this is true or false, but is it true that the human genome was just recently, less than a decade, finally fully mapped?
Okay, so this is a good question. What does it mean to sequence a genome? And we'll talk about that a little bit more in class in a couple of months. Suffice it to say that no genome can ever be fully sequenced, with rare exception. Bacteria, yes. My favorite worm, yes. But something the size of the human genome, the sequence is never completely determined. And that's for many reasons. One, just trivia. Do you know whose genome was sequenced? It was 2004, I think. The human genome sequence was published. That is the genome sequence of the very first humans, not the very first, not Adam and Eve or Neanderthals, but the very first individual whose genome was sequenced, 2004, so over a decade. Who was it? So, right, it depends on who you ask. So it's random DNA samples, and it was actually a combination of multiple individuals that were put together to generate the first human genome sequence, which means the human genome sequence that we look at when we ask, what does this DNA sequence match, and it says the human genome, Y chromosome, it's not a single person's genome sequence that it's looking at. It's a combination of lots of people's genome sequences. So we don't really know who, it's fictional. The reference human genome, the thing that we would call this is the genome sequence of a human, it's made up.
But that's ideal, because if you're using one person.
It is ideal, and that's why they did it, because you don't want to have the genome sequence of one person because it's not representative of everybody. So that's why the initial genome sequence of a human was multiple humans all combined together. So we get what's called a consensus sequence. So you sequence 10 different individuals' chromosome, and you say maybe at every one spot on the chromosome there's one difference in one of the individuals. You say, oh, that's not normal for humans. You say, okay, the other 9 individuals all had an A there. So we're going to say in the human genome, that nucleotide on that chromosome is an A. End of story. Then you go on to the next nucleotide and say, what's the most common nucleotide found in humans at the next nucleotide on the chromosome? You decide that. So the real human genome sequence that's in the database probably doesn't actually exist in any person on Earth, nor probably did it ever. That's why it's called a reference genome sequence. Since then, of course, lots of people have had their individual genomes sequenced. And those we can say for certain, those are that person's DNA sequence. Yeah?
So every human man has that sequence, identical to each other, with few mutations?
Yes. In broad strokes, yes. In subtle strokes, we'll have to talk about what it means to be male, which is a whole other conversation aside from SRY. So there are people that don't have Y chromosomes that have the SRY gene. So they could develop as males, or we would call them phenotypically, if you look at them, male, even though they don't have a Y chromosome. So then we can talk about transgendered and the variety of sexes between male and female. It's a continuum.
Isn't that like a 1 out of like 20,000?
It's pretty rare, but it's not uncommon then. I mean, in a town of 20,000 people, you might expect to find somebody with that frequency then, so.
Can we use this on the exam?
Use BLAST.
Yeah, blast.
I hope you will. Like I said before, way before, that's why I like having everybody have a computer or tablet in the classroom, is that we can use online tools to do real things that practicing geneticists do, and not just made up things that are easy for me to give you on a piece of paper that you can do by hand. I don't even know what by hand means anymore. So at this point, I can send out the practice exam to you. You don't need to work on it now, I'm just gonna distribute it now. And we've got about 10 minutes for questions. So if anybody has any questions now about anything we've done to this point, this will be exactly the same thing we'll do on Wednesday. I'll stand up here, you ask me questions. But if there's anything that's bugging you particularly right now, it's a good time to ask. There will be office hours today, right after class for an hour. Wednesday also, so it'll be another second hour of discussion in my office after the review session on Wednesday. And please do remember that I don't do office hours on Fridays, it's just Mondays and Wednesdays. Sorry for those of you that followed me over to my office last Friday and I had a meeting scheduled at noon, but Mondays and Wednesdays, right after class, office hours. So, there's your practice exam. And please remember, and then I'll open it up for questions, to do these things and prepare for Wednesday. Okay, so questions. Anything at all? Class content, class mechanics, go ahead.
This practice exam, how related or similar would it be to the actual exam?
I wrote some answers to that general question on the practice exam, so you'll see some instructions. But these are questions that you submitted. Thank you very much for those of you that did. It actually inspired me to ask some rather interesting questions on the actual exam, in a good way, not in a bad way. It was nice to see that a lot of you had really thought deeply about what sort of questions I might ask. Unfortunately, a lot of the questions that were sent in were multiple choice questions, which I don't ask a lot of on exams. So there will be some on the practice exam, but basically the question types on the practice exam, I picked those questions that I put on that thing I just sent you because I think they'll make you study things that are important, key topics that we've talked about from the start of class until now. Not because they're formatted in the ways I will ask questions. So it's the content, it's not the actual format of the question that's important. And for those of you that sent in questions that were obviously gleaned from other sources on the internet, I did not consider using those at all. I wanted you to make up the question, not screenshot a question that's on some genetics exam practice website somewhere. It's not, these weren't for points anyway, so that's okay. But the goal is you engage the brain and come up with a question. That's what I was hoping for.
Okay.
Yeah.
Like this very first one where it's the rearrangement of
Yeah, so rearrangements of chromosomes.
Obviously you're gonna have multiple different answers that can potentially be correct.
There are going to be lots of different types of questions that could have multiple answers. Right?
So are you looking for
Be creative.
see the logic is?
I want you to show your work on the exam on Friday. But yes, the logic is more important than, sometimes more important than the answer. There are going to be a lot of questions that could have multiple answers, like chromosome rearrangement questions. And that's fine. Don't worry about it. Sometimes there isn't a right answer. Keep it down, please. You can leave if you want to, because now we're just doing questions and answers that might be very useful for you at the exam on Friday. If you want to talk, get out.
This past weekend, what I did was, because I didn't feel confident in thinking that I didn't know the material, I'm about 75% there, knowing literally everything that we went through.
Mm-hmm.
And I think that the hard part is to understand what you're actually looking for on the homework. But
Folks, please. Will you, if you'd like to talk, you can go outside if you like.
would it be beneficial, because what I did was I went through all of your up to like 2.0 and just
Yep.
re-listened to everything and took notes all over.
Wow.
So would you recommend that or just show up and grow up what I know?
Ooh.
I mean,
I've seen this before on exam days, and I hate to see it. I'm not looking for regurgitation of fact on the exam. So it's not useful. I like to see logic, but if you just start writing down vocabulary that seems relevant, that's not going to earn partial credit, for example. So it's more important to be able to actually do by yourself the things that we've done in class, similar types of questions. You've seen the structures of a lot of questions that will show up again on the exam. So it's more important to be able to do those yourself, maybe in new situations. And I know this is all very ambiguous because you haven't seen an exam from me yet. That's why you get to throw out your low exam grade. So if nothing else, consider this on Friday to be a practice exam that's actually worth points, but if it happens to be your low exam, then it gets thrown out at the end of the semester. It's supposed to be, I know it's not, it's supposed to be a relatively stress-free way to get to know my exams. Okay, one more question, yeah.
The number of questions that you have on here, is that,
Number of questions on the exam is also not reflective of what the exam on Friday will be like, but it will be a 50-minute exam on Friday. Yep, small number of questions. Note, how many questions is the exam worth? I know it's a good question because I asked it. No, how many points is the exam worth? Somebody have the syllabus out? Like 15 points or something like that? 12 points? So it's not, I don't do fractional points, so it would be a maximum of 12 or 15 questions then. Or less.
Have you in the past done like one problem that's got like 3 parts to it, or would all each part be like problem 1 part A, B, part B?
Here's, Fatima, I'm sorry to have to tell you this, but I believe in very flexible grading. When I write multiple part questions, if you, at least, we'll have a discussion later. When I write multiple part questions, if I do, if you screw up on the answer to the first one, but you get the logic to the second and third parts correct, I don't count that against you. I say, okay, if you made a simple mistake early, that doesn't mean you got parts B and C wrong also. If you did it right, given your mistake in the first part, if you did everything else right, then you get 2/3 of the question right, or something like that. So I'm looking for evidence that you understand the material, not necessarily that you always get the right answer, right? So office hours now, I'll see you next class, and we'll continue talking about the exam more.